In September 2024, artificial intelligence finished analyzing DNA samples from graves at Jamestown, Virginia, America's first permanent English settlement. The researchers expected to find evidence of malnutrition and disease. What they found was so disturbing that 3 members of the team initially refused to publish it. The AI identified genetic markers that should not exist in 1607. Mutations that take thousands of years to develop appeared in a single generation. DNA damage patterns that matched no known disease or environmental cause. And most terrifying, evidence of systematic genetic changes across multiple unrelated individuals that suggested something in Jamestown was rewriting human DNA in real time. The settlement, where 80% of colonists died in the first year, didn't just kill them, it changed them at the genetic level. And the AI found proof in their bones. Jamestown was hell. The 104 English colonists who arrived in May 1607 thought they were founding a profitable colony. Within 6 months, 50 were dead. Only 38 remained. The survivors ate dogs, cats, rats, and eventually each other. One colonist murdered his pregnant wife, salted her body, and ate her piece by piece before being executed. Everyone knows Jamestown was a disaster. What nobody knew, until the AI analysis, was why it was so catastrophic. Starvation and disease kill people. They don't rewrite DNA. The research team from Johns Hopkins and the Smithsonian Institution had been extracting DNA from Jamestown burial sites since 2019. They'd recovered usable samples from 23 individuals who died between 1607 and 1610, the starving time when the colony nearly collapsed completely. Standard DNA analysis in 2020 to 22 revealed expected findings. Malnutrition markers. Evidence of typhoid fever. Signs of extreme stress. Nothing shocking. Then in 2023, they fed the raw genetic data into a new AI system developed specifically for analyzing ancient DNA degradation patterns. That's when everything changed. The AI identified anomalies that human researchers had missed. The DNA wasn't just degraded from age and environmental exposure, it showed signs of systematic mutations that occurred while the people were still alive. The pattern was consistent across multiple individuals, and it made no biological sense. The first anomaly was in mitochondrial DNA, the genetic material inherited from mothers that mutates at a predictable rate. Mitochondrial DNA is used to track human ancestry because it changes slowly over thousands of years. A typical human accumulates maybe 1 or 2 mitochondrial mutations per generation. The Jamestown colonists had 15-20 mutations each. This should be impossible. You don't develop 15 mitochondrial mutations in a few months. You don't develop them in a lifetime. You develop them over 10,000 years of generational inheritance. But these mutations weren't inherited. The AI confirmed they were acquired, meaning something in Jamestown was causing mutations at 15 times the normal rate while people were living there. But here's what made the research team stop their work for 2 weeks. The mutations weren't random. Random mutations appear scattered across the mitochondrial genome. These mutations clustered in specific regions, genes responsible for cellular energy production and stress response. The pattern suggested targeted damage, not environmental randomness. The second anomaly was telomere degradation. Telomeres are protective caps on the ends of chromosomes that shorten as you age. They're essentially your cellular clock. Every time a cell divides, telomeres get shorter. When they get too short, the cell dies. Normal aging shortens telomeres gradually over decades. Extreme stress can accelerate it. Starvation accelerates it. The researchers expected shortened telomeres. What they found was telomeres shortened by 40-50 years worth of aging in less than 3 years. Men who arrived at age 25 had the cellular age of someone 70. Women who arrived at 30 had telomeres matching 75-year-olds. This level of accelerated aging shouldn't be possible from starvation alone. Something else was destroying their cells at a rate that defied biological norms. The third anomaly was the strangest. The AI identified methylation patterns, chemical modifications to DNA that control which genes are active, that matched no known human population. Here's why this is terrifying. Methylation patterns are shaped by environment and lifestyle. They're epigenetic, changes that don't alter the DNA sequence itself, but change how it's expressed. If you grow up in a cold climate, your body methylates genes related to fat storage differently than someone from a warm climate. These patterns are stable and predictable. The Jamestown colonists had methylation patterns that didn't match 17th century England, 17th century America, or any modern population. The patterns suggested their bodies were responding to environmental stressors that human beings had never encountered before, or at least not in the genetic record we've found. The AI flagged 47 genes with abnormal methylation. 12 were related to immune function. 8 controlled stress hormones. 15 regulated cellular repair mechanisms. 11 had no known function. They're genes that modern humans carry but don't actively use. Evolutionary remnants that stopped being expressed thousands of years ago. Those 11 dormant genes were active in Jamestown colonists. Archaeologists found the butchered remains of a 14-year-old girl in a Jamestown cellar. Her skull showed cut marks from a knife or cleaver. Someone had removed her face to access the brain. Historical accounts confirmed that colonists resorted to cannibalism during the starving time. But this was physical proof. When the DNA team analyzed her remains in 2024 using the new AI system, they found something that made the genetic anomalies in other colonists make sense and made them worse. Her body showed signs of prion disease. Prions are misfolded proteins that cause brain degeneration. Mad cow disease is a prion disease. So is Creutzfeldt-Jakob disease in humans. Eating infected human brain tissue spreads prion diseases. But here's what the AI revealed. The girl's prion infection had a signature that didn't match any known prion disease. The protein structure was similar to Kuru, but different in ways that suggested a novel prion variant, one potentially more aggressive and more transmissible. And the genetic changes the AI found in other colonists, the accelerated mutations, the telomere destruction, the strange methylation patterns were consistent with prion disease progression. But Kuru takes years to develop. The Jamestown cannibalism happened during a 3-month period in winter 1609 to 1610. Prion diseases have long incubation periods. You eat infected tissue, and symptoms appear 5, 90, 10 years later. That's how prion diseases work. Except something in Jamestown made them work differently. The research team went back to environmental data. What was unique about Jamestown that could cause accelerated mutations? And that water was toxic. Modern testing of Jamestown site soil and sediment found extreme levels of arsenic, lead, and mercury. Not from modern contamination, from natural geological sources concentrated by the brackish water system. The water the colonists drank contained heavy metal levels 50–100 times higher than EPA safety limits. Heavy metal poisoning causes DNA damage. Arsenic interferes with DNA repair mechanisms. Lead disrupts cellular function. Mercury damages neurons and can accelerate protein misfolding. The colonists weren't just starving and sick. They were being slowly poisoned by the water they drank every day. But here's where it gets worse. The AI analysis suggested that the combination of heavy metal poisoning, extreme starvation, and prion disease created a synergistic effect. Each factor accelerated the damage caused by the others. Heavy metal poisoning weakened DNA repair systems, making cells more vulnerable to mutation. Starvation forced the body to cannibalize its own tissues, spreading prions more rapidly throughout the body. Prion diseases disrupt cellular function, making heavy metal poisoning more toxic. The AI modeled this and predicted that colonists exposed to all three factors would experience DNA damage rates 15–20 times higher than normal, exactly matching what the genetic analysis found. In the burial sample set, one individual stood out, aged approximately 30 at death in 1624. He arrived in 1610, after the worst of the starving time, and survived 14 years in Jamestown. His remains showed he died from injury, not disease. His DNA should have shown the same damage as others. It didn't. His telomeres were shortened, but only by 10-15 years of equivalent aging, not 40-50. His mitochondrial mutations numbered 4-5, not 15-20. His methylation patterns were abnormal, but less extreme. He showed evidence of heavy metal exposure, but somehow less cellular damage. The AI identified why he had a rare genetic variant in a gene called GSTM1. GSTM1 produces an enzyme that helps the body detoxify heavy metals and environmental toxins. Most people have functional GSTM1 genes, but some people have a deletion. The gene is missing entirely. And about 5% of people have a duplicated, hyperactive version. This colonist had the hyperactive version. His body was detoxifying arsenic and lead faster than other colonists. Not fast enough to prevent damage completely, but fast enough to survive 14 years in an environment that killed 80% of people in months. But here's the disturbing implication the AI calculated. If everyone in Jamestown had normal GSTM1 function, the death rate should have been 80%. If half the colonists had hyperactive GSTM1, the death rate should have been 50 to 60%. It wasn't. The AI's conclusion: most colonists probably did have protective genetic variants. They died anyway. The environment was so toxic that even genetic advantages couldn't save most of them. Only the absolute genetic outliers survived, and even they showed massive DNA damage. What this means for the historical record. The story we tell about Jamestown is wrong. We say the colonists were unprepared, lazy, and poorly led. They chose a bad location. They didn't plant crops. They antagonized the Powhatan tribes. They made bad decisions that got them killed. All of that is true. But it's not why 80% died. They died because they were being poisoned at the genetic level by water so contaminated it was rewriting their DNA. They died because starvation forced them into cannibalism that spread prion diseases their immune systems couldn't fight. They died because the combination of environmental toxins, starvation, and disease created a feedback loop of cellular destruction that overwhelmed human biology. No amount of preparation would have saved them. Better leadership wouldn't have mattered. More food supplies would have delayed death, not prevented it. The location itself was incompatible with human survival, given 17th century medical knowledge. The colonists didn't fail. They were placed in an environment that made failure inevitable, and they didn't understand why until it was too late. The final analysis the AI performed was trajectory modeling. It took the genetic damage patterns from colonists who died in 1607 to 1610 and projected forward. If these people had survived, how long would they have lived? Average projected lifespan, 8-12 additional years. Even if they'd gotten food, even if they'd received medical care, even if they'd left Jamestown, the DNA damage was permanent. Their cells were already programmed to fail. Telomeres that short don't regenerate. Mutations that extensive don't reverse. Prion infections don't cure. The colonists who died in 1607 were the lucky ones. They died before the worst damage occurred. The survivors of the starving time weren't survivors. They were the walking dead. Their bodies would fail within a decade, regardless of what happened next. One colonist journal entry from 1611 described feeling aged beyond my years, as if God has stolen my youth. He was 28 years old. The AI analysis of his remains shows his cells had aged 45 years. He was right. Something had stolen his youth. Not God. Heavy metals, starvation, and prion disease working in concert to destroy him at the molecular level. He died in 1618 at age 35. His bones showed cellular age of 80. The Jamestown DNA analysis isn't just history. It's a warning about what happens when human biology meets environmental extremes it's not adapted for. Climate change is creating new environmental stressors, extreme heat, novel pollution, ecosystem collapse, and potential food insecurity. We don't know how these factors interact or what their cumulative effects might be on human DNA. The AI modeling used for Jamestown is now being applied to modern populations exposed to severe environmental contamination, areas near industrial sites, regions with extreme pollution, populations facing combined environmental and nutritional stress. Early results suggest we're seeing similar patterns. Accelerated aging in populations near heavy industry, unusual methylation patterns in children growing up in highly polluted areas, cellular damage rates that exceed what individual stressors should cause. We're not at Jamestown levels yet, but the AI can detect these patterns decades before symptoms appear. The Jamestown colonists didn't understand what was killing them until it was too late. We have the tools to see it coming. The question is whether we'll act on what the AI is telling us or wait until we're writing journal entries about feeling aged beyond our years and wondering what's stealing our youth at the genetic level. The colonists buried in Jamestown thought they were building America's future. Their DNA tells a different story, a story of biology under assault, genetics rewritten by environment, and human bodies breaking down at the molecular level faster than anyone watching could comprehend. The AI decoded their DNA 400 years later and found the evidence written in their bones. What it revealed wasn't just how they died, it was how slowly and horribly they were already dying while they were still alive. And the same tools that revealed their genetic catastrophe are now finding early warning signs of similar damage in modern populations. The past isn't just prologue. Sometimes it's prophecy.